Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. This week in Virology, the podcast about viruses, the kind that make you sick. Hello, Brisbane. Woo! That's it? That's all you can do? Let's try it again.
I think so.
Hello, Brisbane. All right. Much better. From Microbe TV, this is TWiV. This Week in Virology, a special episode recorded at Options XII for the control of influenza in Brisbane, Australia. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. I am not in Brisbane tonight, but joining me from Brisbane, Kathy Spindler.
Hello, everybody. And although it's night for Vincent, it's morning for us here in Brisbane, and it's 71 degrees Fahrenheit, 21 degrees Celsius, and partly sunny, partly cloudy. It's been beautiful weather while we've been here.
It's here at 17C. It's nighttime. Also joining us from Brisbane, Steph Langle.
Hi, Vincent. Hi, everyone. Really good to be here. The weather is the same, so I won't repeat that. But excited to be the representative from the Immune podcast today. And we're going to get to talk about viral immunology as well.
We have two guests who are at the meeting from La Trobe University. Stephanie Gras, welcome.
Thank you. Thanks for having me, and hi, everyone.
And from the University of Colorado, Jenna Guthmiller. Welcome. By the way, folks, if you enjoy these science programs, we'd love your support to keep them going. Microbe.tv/contribute is where you can find different ways that you can help us out. We're going to talk about the science that Steph and Jenna are doing. But first, we'd like to know a little bit about your training. Tell us where you're from, your education, and how you ended up where you are now. Let's start with you, Steph.
All right. So I've done my PhD in France, and I've trained as a structural biologist, looking at X-ray crystallography to understand the atomic structure of proteins. And during my PhD, I read some articles from Peter Doherty, Catherine, Steve, and Nicole in Melbourne working on flu, and I thought that was a good place to go for a postdoc. So during my honeymoon, I actually came to Australia, and I had my job interview, signed my contract, went back to France, packed everything, and moved to Australia. And it's been 18 years and never left.
I mean, I have to ask you, is the cheese and wine there as good as France?
No. But it is great, actually. The wine in Australia is really good. The cheese maybe not as good as France, but we do have very good wine.
Wonderful. So then we will get the background from Jenna. Jenna, can you tell us about yourself?
Yeah, so I'm originally from the U.S., originally from the state of South Dakota. Did my undergrad at South Dakota State University, actually studying influenza D, which at the time was actually still considered influenza C-like. Went on to do my PhD in cellular immunology at the University of Oklahoma Health Sciences Center. And then really interested in humoral immunity, antibodies, B cells, and was really excited about sort of these pioneering developments in understanding where antibodies are binding to, how different vaccines, infections drive the specificity of antibodies. And so as a result, I went and did a postdoc with Patrick Wilson at the University of Chicago. He was the one that really pioneered this technology in humans, and lots of really cool work understanding how our past immunity shapes future immune responses. And since then, I've started my own faculty position at the University of Colorado Anschutz Medical Campus. So basically just stayed in the center of the US.
Well, and Jenna, we have a bit of history, not that we knew each other back then, but we share an experience of milking cows. And I want you to tell us your background and how that relates maybe to the current situation.
Yeah, definitely. So I grew up on a dairy farm. So when this bovine influenza case, as I think Andy Bowman said earlier, that there were no bovine influenza experts at the time, or when this really broke out. But it really was an interesting time as somebody that knew what dairy farms were like, how the milk process worked. It was really interesting to hear about, how is this transmitting via mammary tissue? That's weird. But also the fact that when people were getting infected, the few cases that we've had directly associated with dairy farms, that it's happening in the eyes. And a lot of people are like, how is this happening? Like, well, your face is at the same level as, the cow udders, right? So to me, I was like, yeah, that makes a lot of sense that they're getting infected in the eyes. So in that sense, I think it's been really fun, but I think also at the same time, the dairy farm that I grew on, which was very common, in the 90s and so forth, were these really small dairy farms that were, family generational farms. And, I grew up in an area where there were, all of my parents' friends were dairy farmers. And by the time I graduated high school, it was my parents and one other family friend that still dairy farmed. And really, it's become these sort of massive dairy farms. And the way that dairy farming works has really evolved. And I think there's a lot to learn about how dairy farms evolved and how that maybe results in outbreaks like the current one going on in the US.
We'll be excited to hear more about that later on the podcast.
So, Steph, it's only somewhat confusing to have two Stephs on, right?
It is, a little bit. You can call one of us Steph and one of us Stephanie. We'll leave it up to you, Vincent, what you think.
Maybe Stephanie Gras, right? Is that okay? Okay. All right, let's talk about science. Let's start with you, Jenna. And we're going to focus on B cells with you. And I want to start by asking you about immunodominance in the immune response against influenza. What is that?
Yeah, so immunodominance essentially just means that we don't target all parts of a virus equally. So despite that we see all these different parts, our immune response, and particularly what I think about B cells and antibodies, tend to target some parts of the virus more than others.
Right. And in regards to immunodominance, when we think about the targets for vaccination, that has a pretty large impact on what we want to target for broader protection. Would that be fair to say?
Yeah, I mean, I think immunodominance plays a huge part in, vaccine effectiveness and maybe the limitations of how well the vaccines work, because for whatever reasons, we tend to target our antibody response against the parts of the virus that like to mutate.
Right.
And that certainly creates a barrier for better, broader protection against influenza.
Right. So I think mechanistically, do we understand why antibodies against HA, the hemagglutinin protein surface, are variable and are overrepresented? Do we understand mechanistically how that happens?
Yeah, so I would say up until a few years ago, we didn't have a really firm understanding of mechanistically what drives immunodominance. Right. One sort of prevailing hypothesis as to why this is occurring is that if you sort of mutate these epitopes to evade, and epitopes meaning just the specific parts of the virus that are being targeted by antibodies, that if you target those, the virus evolves to get around those, that essentially now you've created new epitopes.
Right.
And what happens is when we have sort of new things that pop up, that we do actually target antibodies against those new epitopes. But it's kind of this vicious cycle that if we mount responses against parts that mutate, it's going to mutate. generate new responses against them, it mutates. And so it's just kind of this ongoing cycle to get around those.
Right, right. And technically, can you explain how are we detecting those antibodies? What are the things that you use in your lab to detect antigen-specific antibodies?
Yeah, so certainly there's a few different ways. So to get at antigen level, right, we mostly do something called an ELISA, so which is a way to measure antibodies in a plate. But to get really down to the specific parts of the virus and on the hemagglutinin protein, there's a few different things we can do. One is that there are antigens that we can make that are, let's say, the head domain of the hemagglutinin protein, which is sort of the more immunodominant domain of the protein. We can also make the stalk or the stem only sort of antigens and test those. And then we can actually start to get down to epitope specificity. So for instance, we can mutate an epitope and see how much that reduces sort of antibody binding, right? Which is an indication of if you lose binding, that suggests there was a significant chunk of that antibody response against that epitope. And then certainly we can also start to get into structural approaches to actually really get into the nitty-gritty of the specific parts of the virus that are being targeted by a single antibody.
Right. And there are other things looking at affinity. You could, for example, are you doing things in your lab where you're rolling over your biological fluid on like a BLI, for example? Could you speak to like the kinetics of binding and how you test that?
Yeah, certainly. So we can measure affinity via a few different mechanisms. You highlighted one, something called biolayer interferometry, which is essentially you have your antigen on a little glass tip, and then you mix that with your antibody, and you can measure how well that comes on and comes off via basically diffraction of light. Right. And we can also use approaches like SPR or surface plasmon resonance, which is a very similar approach. Really, light diffraction is the metric.
Right. Right.
Jenna, does immunodominance happen both after infection and vaccination?
Yeah, absolutely. So some observations that I made when I was a postdoc in Patrick Wilson's lab was we had essentially these really beautiful cohorts of people that had been infected versus vaccinated. We just wanted to compare what were the antibody specificities. And what you find after vaccination is, again, sort of this very stereotypical primarily antibody response against these immunodominant epitopes of the head domain of hemagglutinin. But with infection, what we find is that a lot of that antibody response is to conserved internal proteins, something like NP. And just one thing to note with flu vaccines versus infection, the flu vaccine is really standardized for HA content or hemagglutinin content, but not really the other components of the virus. Whereas when you get infected, it's obviously kind of a crapshoot of the antigens that you're going to see. And for whatever reason, when we get infected, we love making antibody responses against those conserved internal proteins, which really don't mediate protection and are kind of a red herring for our immune system to target.
So from the virus's standpoint, what are the practical consequences of the immunodominance?
Yeah, certainly. I think flu, I don't want to give it too much credit for being smart because it's really kind of just a dumb virus and it just mutates to get around things, but it's figured out how to do that very efficiently. And I think this phenomenon of immunodominance against these antigenically variable sites is something that it favors. Somehow it allows for a lot of mutational tolerance in those epitopes. And as a result, if we're primarily making our antibody response against that and it can mutate to get around that, it's going to maintain sort of a foothold in the population and survive.
Interesting. And so then is there antigenic variation in the stem? So we talk a lot about HA being immunodominant, but what about stem?
Yeah, so there's not a lot of antigenic variation. And what I mean by that is that the sort of parts of the stem or stalk domain that we target, that it's very well conserved from an antibody targeting perspective. The mutations that do come up in the stalk or stem domain are really to improve the function of the HA. So either to stabilize it, to improve, fusion. Right. Those are the sort of mutations we come up. And I haven't really gotten into this so far, but certainly when we think about broader, better vaccines, the stalk or the stem is a very attractive target for that reason. We find that, it doesn't seem to really mutate to get around our antibody responses. And in fact, when people have tried to study, how it could, it's a really weak virus, which sort of implies that it's an Achilles heel for the virus.
Absolutely.
So if you make antibodies to these conserved stem epitopes, how broadly neutralizing are they? Could they hit all influenza viruses? Would a novel pandemic strain be covered, for example?
Yeah, so there's a few different sites on the stalk or stem domain. I'm going to use both terms because I realize the field is a little split on which one to use. So there's a few different targets that we can target on the stalk or stem domain. One of these epitopes we've sort of referred to as the central stalk or stem. And that epitope, if we have antibodies against that, which every single person in this room should have antibodies against that epitope and probably decent levels of antibodies, we know that they provide protection, at least within the group. So either group 1 or group 2 influenza A viruses. We do occasionally find antibodies that can cross between the two groups. And just to clarify maybe a little bit about group 1 versus group 2, group 1 referring to viruses like H1, H2, H5, and group 2 referring to viruses like H3 and H7. And so one observation again is that these antibodies tend to be very group specific in their neutralization capacity. And by having, these antibodies raised by, let's say, H1, that those could be protective against something like H5 that could cause a pandemic.
So what are some of the strategies that can be used to generate vaccines against the stem epitopes?
Yeah, absolutely. So there has been a few different clinical trials actually to try to generate vaccines that are specific for the stalk or stem domain. I was actually able to be part of the crew that analyzed the B cells antibodies after the first one, which happened in late 2017 into 2018. And the premise of this vaccine is that it was the stem or stalk domain from seasonal H1 influenza viruses. And then they used the head domain, so this more variable part, from avian influenza viruses. So something that we had never seen before. We had no pre-existing immunity against that domain. And what was really nicely shown with that clinical trial was that you could sort of increase antibodies against the stalk domain against the epitope that I mentioned previously that we know can provide protection against pretty much all group 1 influenza viruses. So that was a great sort of start to understanding this and proof of concept. And then certainly at the Vaccine Research Center at the NIH, they're doing incredible work to sort of make designer antigens, if you will, that will specifically elicit responses against the stalk domain, as well as sort of take our understanding of B cell immunology, what's the best way to get B cells started and producing antibodies, and really combining those antigens with certain platforms to optimize that sort of stalk-specific response. So I think the future is quite bright for, a universal flu vaccine.
That's good news. That's good to hear. We like to hear that. So curious to hear about long-lived memory B cells to these epitopes. And in this answer, I'd like to hear about, you have a very unique tonsil organoids that you're working with. So I'd like to hear how mucosal immune responses play into the memory B cell response to both immunization and infection.
Yeah, I don't know how much I can say with long-lived memory B cells and those organoids because they're mostly from children. So it's about 5 years, which I don't think is the most accurate. But certainly one thing that has been observed with both my research and then I want to highlight another person's research is that when we get vaccinated and we look even just in the blood, predominantly the IgA response that we see in these individuals tends to be against things from the past, right? Which implies that perhaps these were induced by prior infection and we're calling them by vaccination. And IgA just referring to, antibodies that tend to be induced or sort of selected at mucosal barriers. But another study I want to highlight, and I think this is one of the coolest studies to really show this, not at mucosal tissues, unfortunately, but in blood. There have been these studies also at the Vaccine Research Center where they have vaccinated people who their very first flu exposure was H2N2. And H2N2 circulated in humans from 1957 to 1968. So there's kind of this fine window. And what they find is, and back then people didn't really get vaccinated, right? So any immunity had to be from infection. And what they find is that if you vaccinate these individuals with H2 now, you can get very specific H2 reactive B cells recall of memory, 50, 60 years later, which I think is really incredible and supports this maintenance of long-lived memory B cells, and presumably those that were induced at mucosal sites.
Right. When they were kids, which is interesting that you are looking at those tonsil organoids to try to understand how pre-existing immunity is going to shape their response, just like you said. Yeah, that's fascinating.
I don't know if this has anything to do with what we've been talking about, but I wanted to ask you about flu mist, right? And it's an infectious attenuated vaccine, which doesn't seem to be an improvement on the inactivated vaccines. And I wonder if you could shed some light on that.
Yeah, so I think up until pretty recently, I would be in agreement that I don't think that it was any better. If anything, it maybe was worse than that of inactivated. But there's been some incredible work recently published, but also just at this meeting, largely from Shane Crotty and a postdoc in his lab, Hannah Stacy, where they're showing that by giving LAIV, so the live attenuated influenza vaccine intranasally, that you can see by doing these sort of nasopharyngeal swabs that there are germinal center responses. So B cells affinity maturing against LAIV at a mucosal site. And I think one issue when we trying to compare IIV or inactivated flu vaccines that are given intramuscularly to LAIV, which is given intranasally, we're looking in the blood, but these are fundamentally different routes that the vaccines are being given. And I think we need to have a better understanding of sort of mucosal immunology at the site of infection, vaccination, and sort of come up with immune correlates of LAIV that mediate protection. And just maybe one more thing to add about that is that there's very, very good evidence that LAIV works great in kids. As far as vaccine effectiveness, like the readout of a vaccine, right? But in adults, not so much. And I think there's a lot to be understood as to why that is and how we can potentially generate better mucosal vaccines in adults and also in children, 'cause it's still not great.
Right, absolutely.
So Jenna, I wonder if you'd like to tell us a little about the overview of what your lab is working on. I know you're giving a talk in the next session, so you may not get much chance to catch her right after TWiV, but you can find her later. But some of the things that you do overall in your lab.
Yeah, so I really appreciate these questions because it's been a very good synopsis of what my lab does. So we think a lot about these issues of immunodominance. So how does different exposures shape the specificity of our antibody response? We also think about how different exposure routes, infection, vaccination, what type of vaccine, and how does that shape the specificity of those antibody responses. But then we're also really interested in how driving antibodies against specific epitopes, and sometimes in some seasons, across individuals, that antibody response is very focused to certain epitopes, and we can see that. And understanding how this very focused sort of response can drive the virus to evolve, and what does that mean for the virus fitness and the sort of variants that pop up as a result. And so kind of serendipitously out of that, we've been thinking a lot about how, the antibody response is very focused against the head domain. The head domain is also the site where the virus binds to its receptor. And a lot of what we see is that as the virus tries to evolve around these antibodies, it also changes the way that it interacts with its receptor. And there's really this fine balance of evolving around immunity, but also being able to still recognize and enter into a host. So that's really kind of a synopsis of what we think about.
And with your dairy farm background and your influenza background, have you gotten pulled in? Are there any projects that address some of this?
Yeah, so because we were interested in receptor binding, and certainly with this H5 outbreak, we took this opportunity, if you will, to understand, well, is H5 the current ones that are in cows changing the way that it interacts with its receptor? Because certainly we see an increase in sort of mammals that are getting infected. So is there something changing about it? And so actually I have to run after this to give that talk to show that H5 in North American isolates acquired a mutation last fall. And we're seeing that really increases the sort of glycans or the receptors that that H5 is capable of using, which likely has implications for host range. And certainly I can't say too much about if it's increasing the host range, but it certainly suggests that as you sort of change what you can bind to, that it might have implications for how or what it can infect.
I was fascinated in the last talk that we just heard about the cats in the dairy farms. So do you have thoughts about that and any new plans to address that?
Yeah, not so much with it. Yeah, I know. I sort of feel for the cats because I grew up on a dairy farm. There's always lots of barn cats around. So so heartbreaking to hear about them, getting infected and dying and like not a nice death by any measure. I myself will not be looking into cats, but certainly I have a technician in my lab that's actually applying for grad school right now. I'm going to give away her trade secrets of her NSF application, but she's really interested in these CNS manifestations. And I think, if she gets that and decides to stay in my lab, which I would love, because she's phenomenal, that we could start to look at these things and how, as the virus is changing how it interacts with its receptor, it changes the way that it infects different hosts, tissues, things of that nature, and has different, pathogenesis and outcomes for various animals.
Yeah, and I want to mention you're looking at duck antibodies. Is that accurate? You're very diverse in the types of antibodies you're looking at.
Yeah, so this really came out of a collaboration with individuals at the University of Illinois. With understanding ducks as this really important reservoir for essentially every influenza A virus. And is there something unique about ducks? Is there something cool about these? And so we made this very ambitious grant that was funded by the Howard Hughes Medical Institute through this Emerging Pathogens Initiative to understand duck antibodies, because up to this point, no one has really done that. And certainly it got funded a little bit to our surprise, but very thankful for that. So then we had to find ducks that had been infected, which was a challenge. And I think one thing that I want to highlight, particularly for the options meeting, is that so what we have started doing is getting ducks from hunter-harvested ducks. Okay. But I established that collaboration or that connection actually from somebody that I met at the 2022 options meeting that was doing this cool swine influenza research that I thought was so cool. I talked to her further and she said, Jacqueline Nolting, I don't know if she's in the crowd. And we got talking about bird influenza. And at that point, we had basically just got our notice of award. And I was like, can you hook me up with ducks?
I need some duck blood.
And so I think taking those opportunities to go to those posters or those talks that you're like, this sounds kind of cool. And then just engaging in conversation is so big for establishing new lines of research and, driving the field forward in our understanding of, influenza immunity, but ultimately, different reservoirs and how they mount immune responses.
So, Kathy, that could be a title. Can you hook me up with ducks?
Yes, good thought.
All right, we've been talking about B cells. Stephanie, let's talk about T cells. What is their role in influenza?
So when you think about the immune system, you have to think it's not T cells against B cells, but it's really like a classical orchestra where we have a lot of different immune cells. And it's really important that we actually focus probably on all of them because they all have a very important part to play. So the good thing about T cells is that first they will kill infected cells for the CD8 cytotoxic ones. And also they can cross-recognize different strains of the virus. And that's really important if we were to be able to use T cells in vaccination, because that will probably help us to be protected against different variants and potentially about emerging new variant of influenza.
So if T cells are important for protection, why do we use hemagglutination inhibition as one of the primary measures? And you may have to explain what HAI is for the TWiV audience that's not the same as this audience.
Yeah. So the way we're looking at vaccine efficacy is actually to look at the neutralizing antibody against HA. So HA is a bit like the spike protein of the SARS-CoV-2. It decorates the flu virus and it's on the outside. So the good thing about antibody is that they will actually bind to those HA molecules on the surface and block the virus from infecting the cells. And if you have no infection, you get no replication and you don't get sick. But also it's really good because you don't get transmission, because your virus is just becoming really ineffective. So that's a very potent thing and important thing about antibody. Why we are not looking at T cells efficacy when we look at vaccine, because it's more expensive and it takes more time. So when you're writing grants, because I've been writing on T cells for a long time and be collaborating with people working on antibody and B cells quite recently, it's really cheap to do antibody testing compared to T cells, about tenfold in the price of what we are doing. And it goes way faster to look at antibodies. So it is actually the gold standard to look at vaccine efficacy. Plus, the vaccine, the current flu vaccine, is not really good at activating T cells.
Okay.
And that's an important point, because what does infection do? And does it induce robust T cells? And should that be a goal for a vaccine strategy?
Yeah, it does. So pretty much like Jenna said about the B cells, after an infection, the nucleoprotein of the flu is actually very important for T cells. So proteins which are inside the virus are actually the preferred target for T cells, which is the good thing about those T cells is that the protein inside the virus, usually they mutate less because they don't get this antibody pressure on the surface of the virus. They're also very important for the function of the virus itself. And so it is a bit more tricky for a virus to mutate the internal protein because it will come at a cost of fitness, but also as a functional cost for the virus. So T cells really like the nucleoprotein and also the M1 protein inside.
Right.
So if there's this thing called priming, right, which we haven't mentioned, but would responses to T cells be independent of a priming effect?
To have a secondary T cell recognition. Yeah, so you will get priming, which is your first response to an infection, and that will build actually your memory T cells. Depending on which strain of the virus is infecting for the second infection, it will depend on the level of similarity that those viral infections have with the priming. And the thing is that usually the epitope inside the virus, they don't change as much, and a lot of them will have point mutation that make the epitope really similar, and T cells will be able to actually cross-recognize them and be quite potent most of the time against those new viral infections.
So just as we asked Jenna how technically she measured her antibodies to conserved epitopes, how do we study T cells and T cell responses to influenza viruses?
So there are a few different ways. The gold standard for CD8 T cells is to look at the production of interferon gamma. So you can use ELISpot or you can use flow cytometry to look at the release of those molecules that usually correlate with the level of activation of T cells. Something really important for T cell activation is to be able to map the epitope. So with B cell and antibody, you have the whole protein. So if you use the whole HA protein, you will get a response. For T cells, you really need to know which part of the protein will be recognized. So when the virus is entering the cells, it will be degraded into small particles, small peptide, and those small peptides will be loaded onto HLA molecule. And we all have different HLA molecule, which makes looking at T cells a bit more tricky as well, because you need to have the matching of the peptide that is able to stimulate a strong T cell response and of the HLA molecule as well.
I love the analogy that you used in your talk about how everybody's HLA is as unique as everybody's fingerprint. I thought that was really useful. So just commend you for that.
Thanks.
Do we know anything about specific viral proteins that and what their T cell epitopes might be?
Yeah, so we know a fair few epitopes, but compared to the number of HLA molecule, the number of HLA molecule that we know epitope for T cells and for flu is actually really limited. So there is a massive effort, to my view, that should be done in mapping more of those epitopes to really understand, especially for HLA that are more prevalent in smaller population. So that will be very important for coverage of population if we were to use T cell in a vaccine. So that's something we really need to work on. But the nucleoprotein is really one of the one we know for the moment that has a lot of epitope, and the M1 protein as well.
Okay, nice.
Yeah, and you gave a really excellent talk yesterday. I hope everyone was able to be here for that. It was talking about COVID and HLA types that may be protective or not at inducing T cell responses. So what do we know about influenza and HLA subtypes that are more or less protective? And COVID, you can bring that in too, and how do they relate?
Yeah, so unfortunately, there is not good HLA for all the virus and bad HLA for all the virus, which is probably a good thing for the people who have HLA that are not so great for type of virus. Each different HLA will actually be binding different peptide, and so depending on the virus we are looking at, we'll see different disease association, which if we think at the population level, it is really good because there is not one type of HLA that should be disturbed by all the type of viruses. So we know in flu there are some HLA that are better at stimulating T cells, stronger T cell response. We see some T cells that are highly prevalent against the M1 peptide, for example, that is binding to HLA-A2, where we will see the same T cell receptor again and again and again in individuals that are HLA-A2. And we know that TCR is binding very strongly to the A2M1. It can recognize different variants of that epitope, so that if there is different strain, those T cells are actually able to be activated and to cross-recognize those different strains. And those T cells have long-lived memory as well, so we keep them for a very long time during our life.
Yeah, I like the title of your talk. It was the good, the bad, and the ugly of HLA types. And so for flu, that's also true.
It is. It is for all viral infection. What we don't know a lot about flu and about most of the viral infection, it's the role of HLA class 2 and CD4 T cells. So we know there are some association with poor response to the vaccine for CD4 T cells, depending on which HLA class 2 you're carrying. And so there are a lot of work to do in that space to see if some HLA will actually be better or worse, and that could indicate which part of the proteins within the flu we should probably put or thinking to put into vaccination.
Right, great.
I want to go back to the measurement of or identification of epitopes. So I presume you use T cells from people, but if everyone is different, then how do you ever come to a consensus of what the epitope is?
So usually, there are different ways of doing epitope mapping. It could be you use overlapping peptide, like we've seen a lot of that during the SARS-CoV-2 pandemic, where basically you take the whole protein and you do overlapping 15 mer, 15 residue long peptide. To stimulate T cells, you look at the response, and if you get positive response for your T cell activation, you can look at which HLA those individuals are and go back and see which one will be matched. So the best way to actually say positively that one epitope is bound to an HLA and activating T cells is to either use tetramer binding, so you make the HLA and the peptide, and you make tetramer with a 304 to pull the T cells and show there is binding, or you use antigen-presenting cells that will only express this specific HLA. So that sounds like a lot of work, and for a very long time, it has been a bottleneck of thinking about T cells in vaccination setting. But if you think about the population, some HLA molecules are highly prevalent, and if you take the top 10 HLA in frequency for HLA A, B, and C, you will actually cover more than 100% of the population. So we don't have to focus on the 27,000 HLA alleles we know today, which is a good news. We probably just need to focus on a subset. And if we were to develop T cell vaccination, we should have both the T cell and the B cell, not just one or the other.
Okay, so are these T cell epitopes less susceptible to antigenic drift compared with B cell epitopes?
Yeah, usually they are, because the T cells, CD8 T cells, really like the inside of the virus, and those proteins mutate less, and also because they don't feel the pressure of the antibody on the surface of the virus, they're actually not mutating so quickly.
So is there a possibility for cross-reactivity for these T cell epitopes or T cell responses to other flu types, A versus B versus C, than you might find?
Yeah, so we started looking at this to know whether we can actually have some pan-vaccination type of epitope that will protect against all of them. For the moment, there are not a lot of epitope for flu B and flu C characterized, but there are some, and we've been working in that space. What we've seen for the few examples we have in our hand when we saw cross-reactivity between flu A, flu B, and flu C is that usually flu B is quite good. And when we go to flu C, the difference in sequence for flu C compared to flu A and flu B is higher, and we don't see the same level of cross-protection. And a lot of time, people won't get infected a lot with flu C. So I think it's always also to remember that people will come in contact with flu A and flu B, so they will have pre-existing immunity. And when we look at that, it looks like definitely flu A is most of the time more immunogenic than the two other ones. But there are some epitopes, we found 2 or 3 of them so far, that could have T cell recognizing flu A, B, and C.
That's exciting.
It is.
Yeah, and Jenna had talked about these long-lived memory B cells. And so can you talk about long-lived memory T cells? How long do they last? Do they have the similar protective effect against a pre-existing strain?
So with flu, I find it a bit difficult because we don't know how many times people get exposed and how many times people get vaccinated. When you ask people, did you get vaccinated against flu? The answer is, yeah, I think so. Maybe 1 or twice. And that's where the work we had a lot of question about T cell, fundamental question about T cells and protection in flu for a very long time in my lab. But with SARS-CoV-2, it was actually the beauty of it is it looks almost like a mouse model, but in human population for an immunologist, because we have before and after, and people have been really careful of monitoring themselves. So they know when they get infected, most of them they're testing and they still do, and they know when they get vaccinated because we had to record all of this. And so there are some questions that we are able to ask now about before and after, and how the T cells are actually reacting with the COVID system that we were not able to do with the flu system. But usually T cells keep memory for a very long time. We know if you take the example of SARS-CoV-1, we know from 2003, people were still having some memory T cells 17 years later without having to be vaccinated or reactivation by infection. So we know T cells can keep memory for a very long time, most likely like B cells.
And these studies that you're looking at and others, are they mucosal T cells? Are they systemic? And do you see differences between those?
So far for us, we've been looking at circulating T cells. So we've been only collecting blood. Now we start switching into looking at tissues. From some of the work we've been doing on tissue, they're there. We don't really know if TRM T cells will be playing a role, if we need the circulating T cells to actually come to the tissue place where the virus is when there is an infection, or if we need to block the infection with antibody first, and if some of them escape, we need circulating T cells to actually come and get rid of the infected cells that could be remaining. There is a lot of work that needs to be done in that space.
Sure. And we actually, so in Immune, we did a podcast, I think it was last month, where we talked about that Shane Crotty paper where they were able to get B and T cells from the nasal pharyngeal swabs that were all the way back. They were actually swabbing germinal centers just by that swab. So I think it's really, the mucosal space is an exciting one. So I'm excited to see both of your work in the future.
Some people here might have had the chance to hear Shane talk about that yesterday.
Yes. Yep.
So it was exciting.
So Stephanie, can you talk about some ideas for designing vaccines that induce T cells?
Yeah, so For me, it's really about finding the best epitope and how many HLA we need to incorporate. And the question with all vaccine is whether you have to go with limited epitope or with large part of proteins. So we've been doing some work trying to incorporate some nested peptide between CD8 and CD4 T cells, because if you've got longer peptide, you might be able to actually activate both. And we know if you activate CD4, you will get a better CD8 response, but also a better B cell response, because they're helping the whole immune system to work better. So that's some research we are doing actually in the lab at the moment to try to see if we could actually hit the CD8 and the CD4 in one single go with a limited number of peptide. So that's a possibility of using that to actually stimulate T cells that could be incorporating with the current B cell system.
And you could also use mRNA to encode these epitopes as well, right?
Absolutely. You can use mRNA to encode peptide or to express peptide or part of the protein. I think the really tricky thing is that we do have good platform for vaccine. And I think that we've got multiple choice of how to make vaccine. It's really about which epitope are going to be the best one to be inserted into a vaccine, which I think is the challenge, to be covering the population as much as we can, but also to make sure that, if we come up with a vaccine that people need to take only once, be protected against different variants of different influenza strain, that will be the ideal goal.
So, are there other things that your lab is working on that we haven't touched on that you'd like to tell us about, some of the projects?
So I would say a big question for me, it's always how to activate T cells. What does make a T cell being tickled by an antigen and be a good response that will be actually protective? Because if we manage to understand that, beside viral infection, there is the all other things that T cells do. They're implicated in so many diseases that if we really understand at the molecular level how we can manipulate that response, I think we'll be in a good place to actually use T cells way more frequently in the space of therapeutics.
Okay. One thing that I thought of partway through this is that Jenna hasn't asked Stephanie any questions and vice versa.
Oh, I love that. You should ask each other a question.
I have one question.
What have you wondered about B and T cells from each other?
Yeah, I mean, more so I study B cells because I think B cells and humoral immunity is very cool. But I also will say that I don't study T cells because I think they're too hard. But to be fair, when you pull a B cell out of somebody, if you look at it wrong, it dies. So there's challenges to both. But again, I think this complexity of like the HLAs and the epitopes, it's so complicated. And I just commend you for the work on that because I think, God bless you for doing that because I don't have the mental capacity to do that.
Thank you. Well, it's the same. A lot of T cell immunologists will never dare to think about doing B cell work. But when you start, so I work with B cell and antibody experts, and when you start really thinking holistically about the immune system, I think there is a lot of cross connection between those cells. So scientists should probably do the same and start talking a bit more about how we integrate things and how if you manipulate one part of the immune system, which is T cell or B cell, you actually really influence the other part. So we know with HA, the CD4 response will actually do an imprint on the B cell response, and those two one are talking a lot, and we know CD4 will actually activate CD8 as well. So I think looking at holistically, even if I agree, it's hard to cross between, I would never do B cell. I'm just working with people who do B cell. It's really difficult working as a big team to actually look at those together. It's really beneficial.
Yeah, and maybe just to comment on that, I think when we think about vaccines, which we both were asked questions about this, and I think the approach is that as a person that's trained on B cells, antibodies, and a person that's trained on T cells, we think about things very differently. But at the end of the day, we have to make a vaccine that works for both. Okay. And I think meetings like this and conversations like this are a great way to sort of get the ball rolling on that.
Yeah. And like we saw with Shen's talk, CD4, CD8, B cell, antibody, they're all there, they're all working together, and they all have an important part to play.
Yeah, and we can't throw our innate immunology friends under the bus either. That's very important for everything as well. So, yes.
So Jenna, if you had not become a scientist, what do you think you would have done instead?
I've always been a nerd, always really liked science, so that's always a tough question. I did see this question on there, so I thought about it a little bit. I just like having fun, so I think if I wouldn't have become a scientist, I would have probably gotten more involved in like theater, and, you know, go to Hollywood and just, have fun, joke around, and hopefully get paid for it. But, I guess this works too.
Okay. Would that be musical theater or,
You know, this is a good question. I ask my husband pretty regularly. I'm like, do you think I'd be a good opera singer? Because I have a very loud voice. Like, I think because I grew up on a farm, I had to yell at cows.
Yell a lot.
So I really have quite a good diaphragm. But I don't know, musical theater. I think maybe more comedy. I think I just, you know, like to have fun.
That's awesome. So Stephanie, same question. What would you have been if it was not a scientist?
It's a very hard one because when you do work in academic setting, there's a lot of time you think about, I'm going to leave and do something else. This is, you know, just too hard. But the truth is that it is so much fun. It's such a real privilege to be able to think about a question, be the first one to know the answer. You know, when you think about it, you're in the lab, you're seeing things, and you're just like, oh my gosh, I'm the first one in the world to know that answer. This kind of moment, they're just worth the pen, I guess. Maybe a patisserie chef. I really love cooking, and all my family they are all chefs.
Nice.
And I think about chemistry and making crystal structure a bit the same than making cakes. So maybe that. Maybe
good.
Getting paid for it as well.
I think you've gotten a lot of that in the past, Kathy and Vincent, correct? There's a correlation, I think, between scientists and those interested in cooking and baking.
Okay, also actors. People want to be actors as well. Because as you know, doing science is part acting, right?
Yeah, part marketing, right?
Lots of improvisation.
Do you have any jokes that you want to like try on this audience?
No, I don't. I'm not that good.
Okay. Maybe next time. Yeah.
All right. That is a special TWiV from Options XII at Brisbane. You can find the show notes at microbe.tv/TWiV. And if you have any questions or comments, twiv@microbe.tv. And as I said before, if you enjoy these science conversations, we'd love to have your support. Microbe TV is a nonprofit in the U.S., so your donations would be federal U.S. tax deductible. Go to microbe.tv/contribute. Our guests today from La Trobe University, Stephanie Gras. Thank you so much, Stephanie.
Thanks for having me.
From University of Colorado, Jenna Guthmiller. Thank you so much. Kathy Spindler. Yeah, I did a move for you, you know.
I appreciate it.
Kathy Spindler is an Emerita Professor, University of Michigan. Thank you, Kathy.
Thanks. This was a lot of fun, and I want to thank the organizers of Options XII for inviting TWiV to come. For me personally, it was really great. I've learned a lot. Things that I could have applied in my teaching were I going to be doing teaching going forward, but I've now retired, so that's not the case. But it's been fun to meet people and learn all kinds of new things here, and couldn't have done it without the invitation from Options XII. So thanks.
The things you learn you can use on TWiV, Kathy.
Definitely. We'll be doing that going forward.
Because I understand you're not retiring from TWiV, right?
That's correct.
Yay. Steph Langle, Case, Western Reserve University, at Stephanie Langle on X. Thanks, Steph.
Yes, thank you. Thank you all for coming. This was my first options meeting. I'm really happy to be here. I started in the influenza research community about 4 years ago. And so it's just been a really wonderful community. I'm very thankful for both of you and all the mentors I found in this community. So, and thanks, yes, to the organizers of Options. None of this would have been possible without you and all of you that are here. Thank you very much.
I'm Vincent Racaniello. You can find me at microbe.tv. TV. I want to thank Karen Irwin, Microbe TV's road manager, who you can't miss there. And I'd like to thank the organizers of Options XII for having TWiV, in particular, Kirsty Short, who mediated all the communications. And of course, all of you in the audience, I guess there are about 10 of you, right? Thank you so much for coming. Way more than 10. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.